we've heard a lot about mitochondria already in the conference, and so I won't go into a great bit of detail on the background here. Suffice it to say that mitochondria are complicated. They're dynamic organelles that are constantly moving around the cell, fusing, dividing. They're certainly at the crossroads of metabolism, not only doing ATP, but many other essential biochemical reactions. They're key to the process of apoptosis and cell death. And what I've been really interested over the last 15 years or so is really the role of mitochondria as signaling organelles and how stress signaling in particular is involved in disease, aging, and immunity. Of course, with regard to aging, mitochondria have a long history in aging in terms of the mitochondrial and free radical theories of aging. And sort of the standard version of this is that mitochondria produce damaging molecules as part of oxidative metabolism, the so-called reactive oxygen species and downstream molecules from there. These molecules, of course, are damaging, can damage the mitochondrial DNA, which is inside the mitochondria itself, and cause mutations, which we know accumulate with age. Since that DNA encodes the OXPHOS complexes where the ROS are generated, you can get, over time, ROS-mediated damage to the DNA and to the mitochondria itself. This can promulgate into even higher levels of ROS and eventually lead to cellular energetic crisis, oxidative stress, nuclear DNA damage, cell death, et cetera. And that's clearly a major way that mitochondria probably contribute to aging and age-related pathology. And of course, it's easy to layer upon that other things we know that affect aging, including environmental influences, pathogens, inflammatory states, and other disease states, which can impact this pathway at any of the steps indicated here. Of course, there's a lot of evidence for this idea, and there's a lot of evidence that actually, frankly, is hard to reconcile based on a simple model like this. As we just heard, another major component of aging is inflammation. And Jamie's talk really nicely pointed out that maybe apoptotic cells and the way that our innate immune cells recognize that could be a major nexus for how there is chronic inflammation with aging. Of course, we're still all trying to figure out precisely what are all of the ways that this observed low-level inflammation with aging occurs. And really what I'm going to try to do today is to steal a term from Mark's talk this morning, is look at an edge here, look at some edgeomics and aging, and really look at this intersection between mitochondria and inflammation, and where I think there might be some really important lessons to learn about how mitochondria are involved in aging and maybe think about it in a slightly different way. Of course, mitochondrial DNA, which is sort of the star of the show today, is a 16.5 kilobase double-stranded circular molecule. It encodes 37 genes, 13 mRNAs that encode 13 of the 80 or so OXPHOS components in the intermitochondrial membrane. Of course, to make proteins in a mitochondria, you need ribosomes, and so the mitochondrial DNA encodes 2 ribosomal RNAs. But all the ribosomal proteins are encoded on nuclear genes and imported into the organelle to assemble around those. So there's really 2 things in your cells that comprised of gene products from the 2 genomes, the OXPHOS complexes and the mitochondrial specific ribosomes. The mitochondrial DNA is multiple copies per cell, usually thousands of copies per cell. And down in the lower left here, you can see a DNA stain of a fibroblast. And you can see the sort of constellation of stars out in the cytoplasm there, which is the mitochondrial DNA packaged into protein DNA complexes called nucleoids, which are going to be important for what I tell you today. Just to re-emphasize, mitochondrial DNA is a multi-copy genome, so different than 2 copies of each nuclear chromosome. Mitochondrial DNA is usually present in thousands of copies per cell. Here's 100, so 10 times that for a typical cell. And what I'll emphasize today is that the amount of mitochondrial DNA we have is actually in excess over that needed for oxidative phosphorylation under many circumstances. And I'll give you an example of that in just a moment. Mitochondrial DNA relative to nuclear DNA has fewer DNA repair pathways, so it's unable to repair itself as well as the nuclear DNA. And it also has kind of a weird asymmetric replication mechanism that is damage prone. So I guess one question to think about is, why do we have a multi-copy genome that's sort of prone for damage? And one idea I have is that this is actually a sensor, a genotoxic stress sensor, which my normal talk, I'd go into that a lot more, but in today's short talk, I'm going to stick to another aspect of what's happening here. So mitochondrial DNA, of course, like any DNA molecule, mutations in it, deleterious mutations can be inherited. Mitochondrial DNA is maternally inherited in mammals, and so point mutations, deletions, other disruptions of mitochondrial DNA can cause maternally inherited diseases. These are very complicated because of the multi-copy nature of the genome. I don't have time to get into all of this, but highly affected by the nuclear genetic background, et cetera. But a key point for today is that mitochondrial DNA damage and mutagenesis increase substantially with age. So the story really starts with a protein called TFAM that I've been studying for many years now. TFAM is an abundant DNA binding protein in mitochondria. It's encoded by a nuclear gene. It gets imported into the matrix. It binds mitochondrial DNA. It was originally identified as the major transcriptional activator for mitochondrial promoters on the mitochondrial DNA. But its other main function is really a histone-like function where it bends and wraps the DNA and is really sort of the nucleating events that package the DNA into those nucleoids that I referred to. And TFAM's interesting in that if you knock TFAM out in a mouse, which Niels Larsen did back in the early 90s, you lose mitochondrial DNA and mice die at embryonic day 9 or so. And that's because you lose mitochondrial DNA, no respiration, you're dead, right? So mitochondria important. But what was really interesting is that the heterozygous animals at the time, who really had no phenotype, but he showed that there was at least a 50% depletion of mitochondrial DNA in all of the tissues in those mouse, or all the ones he looked at anyway. And in some tissues, it was more like a 70% depletion. So here's a mouse that basically has half or less of the amount of mitochondrial DNA in all its tissues and is seemingly normal. So I thought that was kind of an interesting scenario to think about and to really ask questions about, why do we need so many copies of mitochondrial DNA? And so a postdoc in my lab, Philip West, took on this project quite a while ago. And he says, why don't we, get some cells out of these mice, do some analyses and see what we can learn. And so these are embryonic fibroblasts from these TFAM heterozygous mice. And you can see on the left here that they have half as much mitochondrial DNA as normal, but that's what we expected. The basal oxygen consumption rates are really maybe down a little bit, but not really a lot, which explains why the mice are not, don't have much of a phenotype. And actually the RNA output of the mitochondrial genome was the same. So the mRNAs, et cetera, weren't being affected. So you don't even need all of the mitochondrial DNA to sustain the RNA output of the genome, at least in this case. But what he did find, if you looked at the cells, is here is that same picture I showed you of the nucleoids in an embryonic fibroblast. The TFAM ones look quite different. I think you can see that the nucleoids, there's fewer of the nucleoids and they appear to be larger. And the mitochondria themselves go into a hyperfused state. So it looks like they're fusing in response to this stress. So we thought this was a pretty interesting scenario to profile in more detail. And what Philip did was global gene expression profiling. And we've done a lot of these types of experiments. This is by far the easiest one to interpret I've ever had, in that the top 35 or 40 genes in these cells that were upregulated are interferon stimulated genes. And if you do the bioinformatics on that, not that you even need to, you just have to read a couple of review articles. The transcription factors that drive that response are the STAT factors, interferon response factors. This is your innate immune system. And we confirmed all of that with RT-PCR of all these ISGs shown above. And we did multiple MES because we thought initially maybe the cells were infected with something and they weren't. But it's actually known at the time, and we had actually written an article with Shankar Ghosh prior to this, that mitochondria themselves are involved in innate immune signaling. And really the entree into that was this MAVS protein, the mitochondrial antiviral signaling protein, which is on the outer membrane of mitochondria sticking into the cytoplasm. And this is really a signaling platform for interferon signaling and for NF-kappa B pro-inflammatory signaling. And what it does primarily, although Jamie would probably argue about this, one of the things it's known to do for sure is that if viral RNAs infect a cell, there are sensors in the cytoplasm to detect that and relay signals through the MAVS complex to upregulate an antiviral innate immune response. There are also DNA sensors in the cytoplasm, which I'm going to talk about here in a minute, and another interesting protein called STING, which is an ER-resident protein that it actually sits at these sites of close interaction between the endoplasmic reticulum and the mitochondria, called the MAM membranes. So the question became, okay, well, we weren't infected with anything. We were messing around with the mitochondrial DNA where we're getting a type 1 interferon response, or what looked like one. So was it this pathway, or was this some novel pathway for activation of interferons from mtDNA? And what we essentially showed, not essentially, this is what we showed, is that it is this pathway. It's the canonical pathway. If you knock out IRF3, which is one of the main transcription factors that upregulates ISGs, the response goes away. If you knock out TBK1, which phosphorylates it to allow it to get in the nucleus, it goes away. So then we backtrack, is it MAVS or STING? MAVS had a modulatory effect on it, but really it was the STING pathway that was driving the majority of this response. And so the STING pathway actually puts you into this DNA sensing arm of the innate immune response. And we're looking for what's the cytoplasmic receptor that's doing this, and it turns out it's this protein cGAS. So cGAS is a really interesting enzyme. It's a nucleotide transferase-like enzyme. What it does is it makes this interesting cyclic dinucleotide called 2', 3' cyclic GAMP. So it's an A and a G that are linked together through some interesting bonds. That's what cGAS does, and it does that in response to binding DNA, usually viral DNA in terms of what was originally identified. But our hypothesis became that this TFAM disruption is somehow leading to the mitochondrial DNA escaping mitochondria, interacting with cGAS and activating production of cGAMP. And what cGAMP does is binds to STING and activates the whole system. So what Philip did was showed in the MEFs and in bone marrow-derived macrophages from the mice that indeed if you do highly purified cytoplasmic fractions that don't have mitochondria in them, don't have nuclear DNA in them, that indeed there is more mitochondrial DNA in the cytoplasm of these cells. So the DNA is getting out somehow. And consistent with that, and consistent with this primed innate immune signature, the TFAM cells themselves were resistant to viral infection. So these are some experiments we did with Akiko Iwasaki with GFP-tagged viruses. And you can see that the TFAM heterozygous cells are resistant to viral infection in vitro. And we also infected the mice themselves with LCMV virus with my wife, Sue Keck, and showed that the mice themselves are actually partially resistant to viral infection. All right, so where does that leave us? That leaves us with that mitochondrial DNA, under some circumstances, can actually escape the confines of the organelle. It can bind to nucleic acid sensors in the cytoplasm and elicit an antiviral-like innate immune response. And in fact, if you do this, you can actually enhance antiviral immunity. Okay, so that's sort of where we started in terms of our aging ideas. I first thought I would show you some new data on watching the DNA actually escape. So this is a cell that's been labeled with an outer membrane, a mitochondrial membrane marker TOM20 in red, and the DNA is labeled in green here. And you can see this nucleoid here is starting off in mitochondria, but then it'll escape over time. So it's out, and now it's moving around. And we've done a lot more on this now. This is actually an old image. It's the only one I had available currently. But as soon as that DNA comes out, cGAS is on it, and we believe the signaling is beginning. All right. So the interesting point that I want to make next is it turns out this isn't exactly an antiviral signal. And here's the main difference that we found. In fact, we knew this at the beginning, but didn't know what to make of it. And the point is that in a typical antiviral response, you get both activation of NF-kappa B and this type 1 interferon response. And usually even when you have a typical interferon type 1 interferon response, you have the interferon genes themselves upregulated. But what we found is that when under these conditions where mitochondrial DNA is being released, there is no increase in interferon genes, and the NF-kappa B pathway was not upregulated. And in fact, we showed that the cells themselves are not, through a variety of experiments, not making interferon at all, or very little. And so that really made us question, well, maybe this is an interferon-independent activation of these interferon-stimulated genes, and so we chased this down. What we decided to do was knock out STAT1 to try to get at this question. And we did that because in a typical interferon response, interferons bind to the interferon receptors. They signal through the JAK-STAT pathway. And STAT1 is involved in all the transcriptional complexes downstream of that upregulate the inflammatory response. So we figured, let's knock out STAT1, and we could say, you know, there's no interferon dependence on this pathway whatsoever. So we expected it to have no effect. Of course, that's why you do experiments. Whoops. When we knocked out STAT1 in the TFAM heterozygous mice and did the similar experiments, you can see very clearly that most of the interferon-stimulated genes, in fact, do come back down. So it is STAT1 dependent, even though it doesn't seem like there's a lot of interferon being made. So we eventually figured this out. Go back one slide. There's a really prominent transcription factor called ISGF3, which is a trimer of STAT1, STAT2 that are phosphorylated with IRF9, and that drives these IRSEs that upregulate interferon-stimulated genes. And it turns out that there's an unphosphorylated version of this ISGF3 factor that activates a subset of interferon-stimulated genes. And in fact, I won't go through all the details. We actually showed that it's this unphosphorylated version of this transcription factor that is mediating this mtDNA response. And interestingly, we also knew there's hundreds of interferon-stimulated genes. We knew we had a specific subset, and that specific subset are the ones that are regulated by this unphosphorylated version. So I'll leave it at that. So we also got interested in whether or not, this is TFAM, heterozygous knockout. It's a weird genetic background, whatever, right? Is there other biological relevance to this pathway? So we started doing some experiments with DNA damaging agents, and we chose the anti-cancer agent doxorubicin. And just to show you some quick data on this, if you treat now wild type cells, forget about TFAM, with doxorubicin, you can see that the mitochondria become elongated, their nucleoids become larger, and they release mitochondrial DNA into the cytoplasm based on our same assays. So phenocopying the TFAM knockdown. Furthermore, if you treat with doxorubicin, you get activation of the interferon-stimulated genes. That's completely dependent on the CGAS-STING pathway and not MABs. Furthermore, if you deplete mitochondrial DNA from these cells with dideoxy-C or ethidium bromide, you can abrogate a large amount of that interferon-stimulated gene response in response to this genotoxic agent. Lastly, if you just target doxorubicin to mitochondria so that you're primarily initially damaging the mitochondrial DNA, that's sufficient to upregulate this subset of interferon-stimulated genes. And we went on from there to show that the TFAM cells are actually resistant to doxorubicin in vivo. And I won't go into all that. We published this in December of 2019 in this Nature Metabolism paper. And there's also some other interesting stuff about how the mitochondrial DNA is leading to enhanced repair of the nuclear genome that I'm not going to be able to talk about today. So where I want to sort of wrap the talk up is since we published that paper and others published similar papers right around the same time, we really have a clear understanding now that mitochondrial DNA can activate many innate immune pathways. So I've just shown you the CGAS-STING pathway, but mitochondrial DNA is known to activate the toll-like receptor 9 pathway, which normally recognizes hypomethylated DNA, of which mitochondrial DNA is. It actually oxidized mitochondrial DNA can activate the NLRP3 inflammasome, and that lead to upregulation of IL-1, IL-1 beta. So multiple ways that mitochondrial DNA can activate the innate immune response. Sort of the other exciting area is that mitochondrial DNA gets into the circulation even, and if it gets into the circulation, now you have a mechanism through which the DNA can activate inflammatory pathways and cause systemic inflammation. So it's really worth thinking about. Also, since that paper, there's been numerous papers that have shown that mtDNA-mediated inflammation is at least involved in some ways with multiple different pathological states. This is from a review article we wrote in 2017, if you want to read more about it. I think a really interesting thing that's arisen is that actually when some viruses infect cells, including RNA viruses like Zika, dengue, and even SARS-CoV-2, this leads to mitochondrial DNA release and priming of the antiviral innate immune system. So I think there's a real fascinating biology here to continue to uncover. So but what about aging? So I'm going to show you some unpublished data now about to ask whether or not this pathway is involved in aging. We're really at the very beginning of this, so this data is very preliminary, but I want to show it to you anyway. So one of the first things is we took tail fibroblasts from young and old mice and cultured them in vitro and looked at their proliferative capacity. And you can see that the fibroblasts from older mice have reduced proliferative capacity in vitro. They induce senescence markers like beta-galactosidase. And interestingly, they upregulate some of the subset of these interferon-stimulated genes, but not the interferon pathway. So that should sound familiar by now. That's sort of our mtDNA signature we defined in the TFAM cells. My graduate student Alva also took human dermal fibroblasts and replicatively aged them in vitro. And here you can see at later passages, we get an increase in senescence markers. We actually get depletion of mitochondrial DNA, which is similar to TFAM. And you get upregulation of a subset of interferon-stimulated genes, but not interferon. So again, there's other things that could be doing this. It could be chromatin fragments, other nucleic acids that could be instigating this. But I think all of this right now has us excited that mitochondrial DNA release may be involved in these cellular models of aging. To that point, if you actually deplete the mitochondrial DNA, kind of like we did with the doxorubicin, you can start to abrogate some of the expression of some of the interferon-stimulated genes, suggesting that it actually is causative. So we have a lot of work to do here, clearly, but I think these are interesting preliminary studies to suggest that mitochondrial DNA release could be contributing to inflammatory states in cells as they age. So to end up here, I will put forward this concept of mito-inflammation. I realize it's a little cheesy, but whatever. Mitophagy, if we can do mitophagy, we can do mito-inflammation. So it's not just the DNA. So as I've shown you, the DNA can stimulate multiple pathways, not just cGAS-STING. Nick Proudfoot showed a couple of years ago that the double-stranded RNA that can escape from mitochondria can also activate innate immune signaling. I mentioned the oxidized mtDNA, NLRP3, TLR9. But mitochondria came from bacteria, right? And so they have many constituents that look like a pathogen. And when those get released during apoptosis or through autophagy pathways, et cetera, those are pattern recognition molecules that can be recognized by sensors in the innate immune pathways. And so I think there could be a bigger role, not just the nucleic acids, but other DAMPs that are involved in age-related inflammation. And of course, with aging comes a degradation or a damage to mitochondria. Pathogens I have shown you, viruses can lead to mtDNA release. And of course, mitochondria are kind of a sponge for environmental toxins and other things. So I think there's a real nexus here where mitochondrial dysfunction and innate immune activation could be involved in inflammatory pathways in aging. And so I'm going to end here. I've shown this slide already, but I'm going to end with my provocative slide, which is a mitocentric view of this idea. So if you really stop and think about all of these things that have been proposed to be involved in age-related inflammation, including apoptosis, as Jamie mentioned, mitochondria are clearly majorly involved in that. But I think there's a mitochondrial component to all of these. So first of all, this increased DAMP production. I just showed you that MTD and other DAMPs can activate the innate immune system. Senescent cells and I think apoptotic cells as well can lead to DAMP formation. other types of signals that the innate immune system will see. And there's a senescence-associated secretory pathway that Judy Campisi and others have elucidated. There's a mitochondrial stress can cause a SASP itself. I've already shown you that mitochondria, that pathogens can actually, bacteria and some viruses can actually lead to the release of mitochondrial DNA. Mitochondrial dysfunction is important for the adaptive immune system. Mitochondrial dysfunction reduces activation and causes exhaustion of T cells, so this could be part of the immunosenescent phenotype. Adipocytes release mitochondrial DNA. There's a couple of papers on obesity causing this. And then the last one is gut permeability and the microbiome. I think there's a real area here to try to understand better is what is the interaction between the microbiome and actually what I would argue is the most successful microbiome, which is the mitochondria, and how that's involved in inflammation. And lastly, I think there's this circulating mitochondrial DNA leading to systemic amplification of these inflammatory signals could really be worth looking into in greater detail. So with that, I'll end, and I guess I end the conference with that, except for the roundtable. And thank you for your attention. I'd be happy to take any questions you have.